Well, thank you very much for that introduction, Amelia. So I arrived here at USF in late 2011, and I was hired as a postdoc in the Paleo Lab by Dave Hollander. So I'm going to be talking a little bit about that work, not everything because of time. So I'm going to exclude things that collaborations like I've been doing with microbiologists, with toxicologists, and benthic ecology. So I'm going to try to concentrate in the water column and interaction with the benthic environment. So for today, I'm going to talk about using organic molecules as tracers of processes of death in the Gulf of Mexico. And this itself is a very large topic. So for today, I'm going to concentrate on a group of molecules that are known as mono and polycyclic organic compounds. And these compounds are very common, very abundant in nature. There are about like 1,000 of these compounds in nature, but just a few hundreds in the marine environment. And here I'm showing just a few examples of very interesting organic compounds that we can measure in the lab. And these compounds are very interesting because, as you can see, and sorry about the typos, it's just this PC doesn't like my Mac format. So the very interesting aspect of these organic compounds is that, as you can see in the screen, they have very different structures. So having a group of molecules with different structures means different functions and different behaviors in the marine environment. So, which means you can, in theory, use them as tracers of processes of death. But you need to understand these compounds, their characteristics, and how they behave in order to put them together and understand the specific process that you're looking at. So, for example, and we look at these compound groups shown here in red. They are very known to be nonpolar, which means they don't dissolve well in water, which means they can stick very well to the lipids in tissue animals, or to particulate organic carbon in the water column, or to sediments on the seafloor. But compounds like the ones in blue now, they're known to be more soluble. So they can, in theory, stick more into the dissolved organic carbon in the water column. So now you have different characteristics that these compounds are behaving very differently in the marine environment. So also, these compounds have very different sources. So the ones shown now in red, they are natural, found in an environment. So they can be naturally produced. For example, we look at sterols. They're very common in the cell membranes of plants and animals. And the degradation products, they can produce other molecules, for example, sterins. Or, for example, the degradation of cholesterol in the gut of large animals by bacteria can produce other sterols like the one shown here. Therefore, this compound group can be used as indicator of anthropogenic influences or contamination of coastal environments through sewage. So also, hopens and sterins can be used to trace organic matter from microalgae and bacteria. And as well, the degradation products of some of these compounds, for example, sterins, can produce odors like triaromatic steroids, which are very well known to be stable in nature, except if they are exposed to sun. Therefore, these compounds can be used to trace photooxidation processes in the marine environment. Also, other compounds like the ones shown in here in yellow, they are polycyclic aromatic hydrocarbons, or PAHs, and they have different sources. They can be naturally produced, or they can be produced through combustion through anthropogenic activities. So also, the compounds now highlighted as blue, they are well known to be very persistent in the environment, while the ones now shown in red, they are very known to be toxic or even mutagenic to biota. So altogether, these compounds can be used to trace processes or sources of organic matter in the marine environment. And that's what is my main objectives of my research, to use the combination of these compounds to understand what is going on at depth in the Gulf of Mexico. So I just want, before I jump into my research, to give some examples of application of these molecules. For example, some of these organic compounds, they can be used to trace bacteria in paleo environments. Therefore, they can be used to reconstruct of early life evolution. Also, other compounds like polycyclic aromatic hydrocarbons, they can be used to trace the origins of organic matter and the transformation processes in environments. Or you can take artificial or anthropogenic created molecules like pesticides and study relative to redox reactions in the environment, so you can understand that fate and transformation products, again, in natural environments. Another example is the use of organic compounds, very different from the ones that I showed you, to look at carbon sequestration in mangroves. And this was a project that I was part of before coming to USF, where the main objective was to look at nutrient limitations not only on the plants, the mangrove trees, but as well the bacteria. So understanding how bacteria and plants are limited by nutrients in the same system, you can use it to better understand how this system are limited in nature, and also you can manage this in terms of possible negative effects or impact by humans or by phenomena like sea level rise. So for today's talk, I'm going to divide it in 4 sections. I'm going to be talking about geochemical indicators, the fate of organic compounds, time series, and the new studies that already Amelia briefly mentioned, and there is some space for future collaborations with other scientists or professors here in the college. So the first section is geochemical indicators of ecosystem change and processes at depth. And this study was conducted in the De Soto Canyon, which is located in the northern Gulf of Mexico. This study was funded mostly by the consortium CIMAGE. And for the people that are new in the college, this is a consortium hosted here at USF and is directed by Steve Morawski. And this is a very large consortium. We are about like 181 scientists, including students from 18, 19 institutions from the United States and other parts in the world. So the main goal in this consortium was to study effects of oil spill in the marine environment. But we didn't look only at the deepwater horizon oil spill effects. We as well went and looked for samples to understand what happened after the Ixto-1 spill, which occurred in the southern Gulf of Mexico 30 years ago. So one of the most striking things of studying these 2 oil spills, which are the largest accidental oil spill in the world still today, both in the Gulf of Mexico and separated by 3 decades, and that for those 2 oils, we still have the same problems and data gaps. So we were faced with the same problems like 30 years ago scientists were faced to, this is incredible. And for example, the first one, we didn't understand very well the hydrocarbon fractionation at depth and what it means. So you see this diagram, this is a representation of an oil droplet. The oil has so many different organic compounds, and as you can see here, again, there's very different structures. So again, it has a lot of different functions and behaviors in the marine environment. And we didn't understand very well how all these different compounds with different behaviors will react when it's released at depth. Also, we didn't know very well about sedimentation redistribution processes. Also, these both oil spills highlighted the massive data gap, especially for the deep pelagic zone. And we didn't know very well the fate of these organic compounds in the marine environment. And we're still working on this, especially to understand what happens with these oil organic compounds related to the marine food webs, especially the interactions with large pelagic fauna. So this was my first study that I did here at the college. And so when I came here, so as I said, I was hired by Dave Hollander, and he gave me the challenge. He said, your job is to demonstrate that oil residues from the deepwater horizon reach the surface of the sediments. And just by that was the big challenge, because we all know oil floats, doesn't sink, right? And that concept is just because we didn't understand very well how organic inorganic interactions affect the transport of organic molecules in the water column. So the second challenge that I had when I started working here was that the sediment cores were collected before I arrived here. So I arrived late in 2011. The oil spill was in 2010. There was a lot of cores that were collected, and they were extruded at high resolution, which means they were sliced, cut at 2 millimeters interval. And this was necessary in order to be able to capture changes in very short periods of time. So what that means for the chemistry part is that you get a tiny sample at the most difficult matrix that you can use in chemistry, which is sediments from the deep ocean mixed with oil. So my first task was to change the chemistry, the chemical methods that traditionally are used to look at chemicals, organic compounds in sediments. And these methods have changed a lot through time, and we adopted different methods from different labs, and we make it our own. And at the end, we were able to develop a one-step extraction and cleanup procedure coupled with a single-run analytical steps, including 160 compounds, altogether as is represented in these different chromatograms. And this is very different from traditional chemistry methods because normally you get your samples, you extract it, you use column cleaning procedure, and you separate some of these compounds in fractions. So, and each of these fractions is run separately in the GCMS. That takes too much sample, takes too much time, money, solvent. So I created one method, and we now can use very tiny amount of sediments and also biota, and we can run and detect these 160 compounds, no interference with known targets, use very little solvent and time usage. Now with the thesis of Thierry Berlet, we are being adding more compounds. We are pushing the analytical techniques, and I think now we're like about 240 compounds that we can run and detect in one run. So. 256. Oh, 256. So she has the exact number. So as you can see, we're very excited about this. So having resolved the chemistry part, now I can show you the first data set. And this is just a section of the first data set, just 3 profiles, just to simplify. So on the top, I have 2 organic compound groups, hopens and sterins, that these are known, and we target the ones that we know that were particulate specific to fingerprint oil spill. So every oil has a different fingerprint, and you can use it to detect where that organic compounds are coming from. So we use specifically these 2 organic compound groups, but also we use stable isotopes. And we use radioisotopes to add chronology in our profiles. So with this data set, we were able to demonstrate for the first time that oil residues from the Deepwater Horizon reach the bottom of the sediments in the northern Gulf of Mexico. So we were very excited about this. And it was basically because when you look at the profiles, you see that there is the largest changes were on the top, which is after the Deepwater Horizon spill. So we saw increase in concentration of those compounds that we know that were coming from the oil. But also we saw these changes in the isotopic composition of the organic matter in the sediments, which is indicating that there was a deposition of larger amounts from algae as well as sediments contaminated with oil. But also when you look at these profiles, there is something that strike out. It's just if you look at bulk measurements, which is the measurement taken from the stable isotopes, you see these different colors are from 3 different sites in the DeSoto Canyon. They're very the same, they exact what you expected from the deep ocean, unless, except on the top, which is this extreme deposition of this material after the spill. But when you look at these hopens, when you look at a specific molecular level or molecular composition, you see there is more noise. And sometimes people are like scared about noise in data, but I personally kind of like it. I think where most of the information is where you see the noise. So there is a lot of differences among the sites using or looking at the molecular level. So I wanted to go deeper into my data set, and I look at very different compounds, and should be moving, okay. And so in this case, I look at polycyclic aromatic hydrocarbons. And here, the 3 sites from the DeSoto Canyon are separated in 2 in separate graphs. The gray bars are showing sedimentation rates. The white dots are low molecular weight pHs which are known to dissolve better in water, while the black dots are high molecular weight pHs that are known to stick more to the organic particulates, to sediments. So with this data set, we were able to demonstrate for the first time the natural processes of depth transported Deepwater Horizon oil residues to the surface of the sediments on the seafloor. So for example, if we look at this site, we see that the increase in low molecular weight pHs that does not have a match with increase in high molecular weight or sedimentation rate. So basically this was indicating that there was a lateral transport of submerged plumes that were formed after the spill, composed mostly of low molecular weight compounds. And so there was the direct contact, but we think it was the direct contact of these submerged plumes on the continental slope surface sediments. When we look at these other sites, the distribution of the profiles are very different. So there is a larger sedimentation when there is an increase in the low and high molecular weight pHs, as well as those increase in signatures in carbon-13, indicating there was a larger amount of algae material deposited on the surface of the sediments. This itself indicates that there was a vertical transport of sinking marine snow that was contaminated with oil. The third site, actually, it was very interesting because it was a delay in the deposition of these organic compounds, and it was mostly from pyrogenic or burned organic matter that we think it was coming from the large burning of the oils on the surface of the water during the spill. So to better explain this, I'm going to use Kendra's conceptual model that she published in 2016. And this is a great summary of what happened to the oil that was released at depth, which shows that part of it went and formed these submerged plumes, and it has mostly contained these low molecular weight compounds, while the rest that went to the surface of the water column and formed these oil surface slicks are composed mostly by high molecular weight compounds. Later, this oil or these compounds on the surface mixed with these algae plumes and sediments, got heavier and sank. And that's what we see in the sediments. We see these sometimes where it was the plume, we see these different chemistry that it was coming from the surface of the water column.
Did they not use a lot of dispersants for the oil as well?
Yeah.
And help us understand where that shows up as it's relevant. Right?
So it's very hard. So I didn't look at dispersant, and also so there's controversy between the use of dispersant or not, but in the surface, what happened is that most of it that is in the surface will dissolve. So in theory, we'll mix more with the sediments and the algae and sink. What we will see a wet drill signature of the chemistry, unless we target specifically the dispersion, but we didn't look for that.
Thank you.
So again, here we wanted to test if what we see in one location in the northern Gulf of Mexico, which is the Desoto Canyon, we could see the same pattern at a larger scale. So the next step, what I did is just took publicly available data that was generated by BP and through the NERDA program, through the government, and I took this data and processed it and divided it in three areas: the coastal, continental shelf, and the deep sea. And what I did with this data was to generate a lot of ratios and distribution of organic compounds. And basically what I did, I compared the surface of those sediments, of that data that correspond to the surface of the sediment, with the layers beneath. So basically to compare before and after the spill. And I calculated a lot of ratios, distribution of all these different compounds that I already mentioned before. And this was for almost 2,613 different sites. We did the same exercise. And don't worry, I'm not going to explain each of these graphs. This is too much. But in general, what we did this exercise for each of these cores, and we generated a spatial model for that, which is easier to understand, easier to compare with other data sets. And basically what I'm showing here is blue are showing the areas where we didn't see a significant amount of deposition of oil residues coming from the Deepwater Horizon spill. The colors from yellow to red is showing the increase in the amounts that was deposited. And what we found is that 50% of the area, or 110,000 square kilometers, were affected by residues coming from the Deepwater Horizon spill. With this, we calculated budgets, we calculated a lot of numbers, but one of the things that I think is more interesting is that with this map, I was able to compare this data with previous data from the water column. And it was very interesting to see that the surface water processes that people have described before in separate studies actually correlates very well with the distribution of deposited oil residues on this large scale. But again, we wanted to understand better if everything is the same from coastal to deep sea, and we did more ratios, and I'm showing just a few here. These are ratios that we calculated, and we compared the different areas, and here is showing the concentration of all those organic molecules on the surface of the sediments, and we compare the surface and the layer before in order to compare before and after the spill for each of the zones. So in general, what we found is that the most recalcitrant, heavier organic compounds were deposited on the coastal areas, and the more toxic were deposited at the deep sea. But also when you look at only the sediments that they were deposited before the spill, you can see for example, to start with the continental shelf, there was not much going on in the continental shelf. So it looks like the natural of the continental shelf is a transition zone between the coastal and the deep sea. And you can think about the Mississippi bringing all those sediments, most of it is in the coastal zone, and there should be processes that move this, not in actually reaching the surface of the sediments in the continental shelf, but moving deeper. And this is demonstrated here by looking at the sediment layers deposited before the spill, which there is larger amount of organic compounds in the deep sea compared to the other areas, especially in the coastal zone, which means that the deep sea sediments serve as repositories of organic matter, and in this case, contaminants as well. And I will bring this concept back because this is very important when you're thinking about the fate of organic compounds in the northern Gulf of Mexico. For example, the second section, which is the fate of organic compounds, and for these, we only didn't look at the northern Gulf of Mexico, but as well the southern Gulf of Mexico. And we wanted to understand if the processes that we observe in the northern Gulf of Mexico, we could actually see them as well in sediments from the southern Gulf of Mexico. But it was tricky because the sediments, we collect them 30 years after the spill. And also we wanted to see if this, or residues, if we could see them, if they have a potential, or CO can have a potential impact to biota after so many years of being deposited in the marine environment. So let's start with the northern Gulf of Mexico. Here are sediments collected in the De Soto Canyon, and now we have total sum of hydrocarbons, concentrations over time, and in the right side we have the composition of organic molecules in those sediments. And one thing to note here is that when I look at the sediment over time, what we do, we use chronology to follow the layer that was deposited on the sediments over time. So over time, that surface layer become buried underneath, and we wanted to follow that in order to be able to see what happened with these compounds. In very few words, there was a big change in the concentration, but not in the composition, which means degradation processes are not driving the concentrations or the fate of these compounds in the deep sea of the sediments. This is something that many of the groups are not kind of taking into account because they always look at these changes and they're saying that degradation is the main process. But if degradation is the main process, you will never see that the composition is the same because, for example, bacteria targets molecules that are easy to degrade. So when that happens, you see that the composition changes, and then you can go and say, okay, this is a lot of biodegradation, this is what is going on, but here is mostly the same. So there might be other processes affecting this, and I will come back for this topic. So now sediments for the deep sea collected in the southern Gulf of Mexico. We were able to collect 22 different cores in 2015, and you can see these transects. These transects were determined based on the distribution of surface oil slicks that were observed in 1979 and 1980. And it was a work conducted by Shoimin and his students. And we used that trajectory of the oil surface slicks to look at these cores. So we did all our chemistry, and also to point out, the yellow star is showing exactly where the XO1 spill occurred in 1979. And we were very lucky to get a permit to get into that exclusion zone to collect sediments just there. And so we did all our chemistry, and we found that 4 of the 22 cores that we collected, so 4 sites have, looks like have significant oil residues from the XO1 spill. Very interesting, we were able to support this information with chronology, but when we look at sedimentation rates, it looks like there was not an increase in sedimentation rate, so other processes might have been moving or transporting these organic molecules at depth. So now I'm comparing the composition of organic molecules between these two areas. So we have the northern Gulf of Mexico sediments 3 years after the Deepwater Horizon spill, and then the southern Gulf of Mexico 3 decades after the XO1 spill. And what we observed are small differences, which indicates, again, biodegradation is a very slow process at depth. So there's a long-term persistence of these organic compounds on the sediments. But we see there's a significant change in this orange color dish bar here, which represents my high molecular weight pHs, which is odd because these compounds don't degrade almost. If they should stay there. So there might be other processes besides degradation. So in this case, we should have another process that we haven't studied yet in depth in order to explain that. So we look other groups and their research, and there was a group looking at oxidation products of organic molecules in shallow waters. And what they did, they were able to look at the polar, very high molecular weight compounds that they're not detected by GCMS. It's very hard. We cannot separate them. And they use this technique called electrospray and run the samples in MTS CMS, which is able to collect that fraction that we cannot look and separate in GCMS. And what they found is that here in green is showing the reference oil, the Macondo reference oil standard, and in red is showing samples taken from Pensacola Beach, with having this higher concentration or relative abundance of oxidation transformation process, and here of the heteroatoms for oxygens, which means things are deposited and they are changing and not necessarily by biodegradation processes. So we wanted to test if this was happening as well in the southern Gulf of Mexico. And so in this case, we went to a very different environment. And I was very excited because a lot of my previous work before coming to USF was in mangrove system. So we had the opportunity to go back there. And this is very interesting because these mangrove forests are in the Campeche area. And we had fishermen that went with us, and this was a field trip in 2016, and kind of like guide us through the forest and told us that they can remember exactly these sites where the picture was taken, that there was a lot of oil in these forests, about like 0.5 meter amount of oil was in these areas after the spill. Still, I don't understand how these forests survive, but there's mechanisms that actually nature can cope with, and I think these mangroves are a special case for that. So in 2010, West Tunnel was able to go back to the same sites, and he found that there were still oil residues at the surface of the sediments. But he didn't collect any samples or did any chemistry. So we went in 2016, and we found, it's hard to see here in the picture, but there are oil like tars on the surface of the sediments. You can see better here on the prop roots or the roots of the mangrove trees. But very surprisingly, we found that there was a 2 centimeter thick layer of oil that was buried on the sediments in these mangroves. And when we opened this to collect the sample, the oil was still liquid and smelled, so it's kind of like fresh. So we wanted to know, well, first of all, is Ixtoc one spill, or could be something else, right? And what is the weathering of this oil? So we collected the samples, we did all the chemistry, and we did the fingerprinting, and actually this is Ixtoc-1 oil that was deposited in these mangroves. But again, I wanted to go deeper. I always wanted to go deeper into my research and understand better these oxidation products. And last year I got an opportunity to run samples in the National High Magnetic Field Laboratory in Tallahassee to look for those oxidation transformation products that we cannot detect here at USF. And here are some preliminary results showing in green the Ixtoc-1 oil reference standard, and in red these buried samples that we collected from the mangroves from the Campeche. And we see there's a large transformation products of oxygen heteroatoms, but also we found there's thousands of uncharacterized transformation products. So there's probably coming from very different weathering processes. So, but when you look at these type of analysis, you get the level of resolution is a heteroatoms, which means I cannot tell anything else from here. And I always wanted to make this data that you can apply to make a health assessment of the ecosystem. So you can say if actually these have a negative consequences to the biota or to the system itself. So for that, we adapted and created a method in the lab to look at oxidation products, specifically of pHs. And in theory, a lot of people think that you cannot do this in GCMS, but when GCMS MS technique is very powerful, and when you target a specific organic group, you can actually do it. So we developed a method, and we were, and we wanted to target these molecules because they are known to be more toxic and more mutagenic than the traditional pHs that normally people look into it. So we went and did that, and what we did, we compared the weathering of oil samples, sediment samples, with these oxidation products, and we found that when samples were more weathered, more affected, they have more of these oxidation processes. And here I'm showing more detail on that. So on the top I'm showing an oxy-pH, which is natural produced in the environment. And then we have these more contaminated, more toxic compounds, nitro, oxy, and hydroxy pHs. On the left side are natural samples like algae, roots, leaves that we collected in the environment. And on the right side are samples that we determined through fingerprinting that they were contaminated with X1 oil from the spill. And what we found is that natural produced oxy pHs, they're very abundant in the natural samples and less in the contaminated samples. And the opposite it is found with the natural oxy and hydroxy pHs. Also, a very interesting thing is that the concentration varies among the type of samples, which means that it depends on the oxidation process, the weathering process that affects the concentration of these compounds. So in other words, that using these weathering products, these specific oxidized pH products, we can use it to fingerprint different sources, inputs in the environment, transport mechanisms, and the fate in the marine environment. And also, we can use this data to do health assessment in the environment for long-term periods of time. Because a lot of times, when you look at reports, they look at pHs and they always say, oh, it's lower or decreasing, but they don't look at the transformation products. And now we can apply this to future studies. So now all what I have been talking about fate is in terms of weathering. But there's another way that you can look at fate. So it's not only what is happening in the system and changing the chemistry, but as well we have the hypothesis that things can be moving in a different way. So, and this is what we think is happening here in the northern Gulf of Mexico. So things that reach the surface and sediment not necessarily can stay there. The bottom of the ocean is very dynamic, and we have been looking in detail on these movements at depth. So last year, I got a grant to study the resuspension distribution of these organic molecules that were deposited on the surface of the sediment. And this is a project in collaboration with, erase all my collaborators. Wow. Okay, so with other, 3 other collaborators from other different institutions. And the idea was to, or our hypothesis was that the area that was affected by the oil residues is getting bigger because things are not staying where they reach the surface sediments, they're moving. And they're not moving physically, like just randomly. They're moving and depends on the specific habitats on the seafloor. So we were able to go last year and collect sediment cores from the Deepwater Horizon sites and going downslope and target those specific areas, looking at very detailed the different environments on the seafloor. So here I'm just showing a very, very preliminary data. It can change later when we put it all together. But basically what what we found lately is what the red dots are showing possible Deepwater Horizon oil residues. And what it's showing is that things have moved from 35 kilometers up to 100 kilometers from the Deepwater Horizon sites. So the area of impact is larger than we thought. And also, but I need to, so the next step is to kind of compare the chemical data with chronology, C14, especially C14, because it looks like there is a very neat opposite correlation with C14. So this is not oil spill. This is what we're using is now these organic molecules around the oil spill as tracers of processes. So what we found is that actually these guys here, it looks like they don't have Deepwater Horizon oil residues, but they have very old organic matter based on C14. And their structure and the distribution at the molecular level is super interesting. But I still need to kind of like put together the sedimentation rates, the suspensions, and multivariate analysis, and do all that stuff to make a better assessment. But overall, what we see is that what we see in the chemistry is very different among the sites, which means things are not just moving downslope. There depends on the topography of the seafloor, but as well the circulation processes at depth. And so also with this project, what we want to understand is the implication of these deep processes in the oceans. Because recent studies have shown that some resuspension events in the northern Gulf of Mexico can move sedimented organic matter up to 100 meters from the seafloor. And this is very large. We thought it was kind of something small, but these can be very large events. So we want to better understand the implications while bringing all this sediment to the water column that in some cases it's contaminated as well. And what is the effects of this natural process bringing back again over time these sedimented material that has contaminants to the water column, and what could be the effects for the deep pelagic ocean. And I'm very interested about this topic, and I hope you understand why I'm so excited about it. And when you look at this slide, which represents just a little bit about the deep pelagic ocean. So this is just an example of the diversity, the richness, the biodiversity, and what millions of things can happen in the ocean that we haven't looked into this. And as I said at the beginning of my talk, the deep pelagic ecosystem represents the largest existing data depth in the Gulf of Mexico, even though it's the largest in the Gulf of Mexico by volume. And just to give a little bit of sense of why this deep pelagic ecosystem is so interesting and cool to study, not only for the Gulf of Mexico, but worldwide, they represents, or they store the largest global biomass and mesopelagic fishes. Also, they links very efficiently the lower and high trophic levels. And if you see in this diagram, there's this arrow here, which is representing vertical migration that occurred in these animals. And not all of them, but most of these animals, they move every day. They move during the night to the surface waters to eat, and then during the day they go down. And with that, they have a lot of dynamics and interaction with animals that only live in the surface or only live in the bottom. So specifically for the deep pelagic Gulf of Mexico, they as well have been demonstrated they're very important for carbon sequestration, fisheries production, and even bioproducts accumulation. So I was very lucky to be part of this amazing team. We got a grant funded since 2015 to look at the deep pelagic nekton dynamics of the Gulf of Mexico. And this is a collaboration of, we're like over 20 PIs from 8 different institutions. And the main goal was to understand the deep pelagic ecosystem of the Gulf of Mexico. Not only to look at diversity, biomass, and species identification, but also to look at genetic variations and also water column dynamics, and of course chemistry. So my role in this project was to look or determine if the deep pelagic fauna was exposed to contaminants during the Deepwater Horizon spill. And this was our first main goal, but it changed. This question changed, and I will show you why. We thought that the main problem was 2011 when the spill was going on in summer 2010. And this is just to show that the Deepwater Horizon spill, to remind everyone, began as a deep pelagic phenomenon. And the profiles, these are pHs, showing that the whole water column was contaminated in 2010, showing in blue, especially at 1,200 meters where the deep plumes were formed. But also, which was very surprising, from what we all thought, in 2011 we could still see high concentration of these toxic molecules in the water column. We don't have more data after 2011 because people thought there's nothing else in the water column, so it's not worth it to even spend the money. But then with this project, I was able to collect samples and to analyze them not only for invertebrates, but as well as fish, mesopelagic fishes. And here I'm showing a data set for octopus and squid. And so what I'm showing here is the abundance or relative abundance of pH compounds from 3 different time periods. 2010, 3 months before the spill, there was a cruise out there collecting these animals, so we were very lucky that they stored them very well. 2011 and 2015 and 16. So by looking at these composition changes in time, and calculated ratios, we're able to see that after 2011, after the spill, the chemical signature of this compound changed to a more petrogenic source, which we make the interpretation that in order to see this huge change in this specific chemistry, it has to be a big event changing that, which is correspond to the Deepwater Horizon spill. But then when we look at the concentration on the right side, over time, we see a pattern that we were not expecting. When you have a one event given a contamination in one side, you expect to see an increase and then a decrease. But in this case, we saw increases, a twofold increase in this concentration in 2015 and 16, 5-6 years after the spill, not after. So what we look into this to understand if you see a change in the composition but not in the concentration, what's happening. And what we think, our hypothesis is that in 2011, because we look at the lipid concentration of these animals, they were very low compared to all the other years. And you know, these compounds, pHs, they stick very well to the lipids in the tissue of animals. So if you have lower lipids, it means you have less material where to attach to. So there's not that mechanism that you can store this molecule, you don't have that much lipids. But what it means to have lower lipids after the spill, not before, not after 4 or 5 years. What we think is that there are 3 hypotheses. One is that we always be able to collect the survivals. So maybe the animals that we were able to analyze were the ones that were less exposed. That's one hypothesis. The second one is that probably there was not much to eat out there. 2011, a lot of the prey for these animals was very little, so they were kind of like not having a lot of sources, so they were kind of skinny and didn't have that much lipids. Or the quality of the food, the lipid composition of the animals changed, there was not that much, and their diet was poor, and they didn't accumulate that much lipids. Their hypothesis, we don't have a way to prove this, but back in 2015 and 16, lipid composition were fine again, compared to the data that we collected before the spill. So what we think is that the Deepwater Horizon create secondary effects to the trophic web that may have influenced the lipid content and therefore the pH's concentration, and that's what we see in this trend over time. Also want to note that we analyzed 7 different species of invertebrates here, and all the different characteristics, thinking about their diet, they were different, but they didn't explain this change over time. Their sizes, their ecology didn't explain this temporal trend. And we knew there was something going on here, not only because of the composition, but because we have the fish pelagic, the mesopelagic fishes, temporal analysis that I'm showing now here. So here I'm showing the concentration of pHs versus time. And if you notice, the time is different, and it's because we were able to get samples from different studies. So now we have samples before the spill, but in 2007, and then we have 2010, 11, 15, and 16. The results shows that the temporal trend that we observe are not explained by any of the biological characteristics from 5 different species of fishes that we were able to analyze. And also what we see, there was an increase in concentration as expected if you have a point source contamination event, and in this case was about 7 to 10 times increase compared to the pre-spill values. But then in 2015 and 16, concentration were back to lower levels. It's hard to tell here, but they were not at the baseline levels, but they're very close. So at that point, we said, well, the system itself is showing there was contamination, but now recovered. But then again, I wanted to go deeper in my data, and I was very surprised because if you see this diagram, we look at very different tissue, and I'm not showing all this data. But one of the most striking things when we look at the eggs, so a hatch egg still in the ovaries of the animals, we found that they were very high concentration. So in 2015 and 16, the decline of pH levels was observed only in the muscle tissue, but not in the hatch eggs, which have 50% higher in concentration. Oh, that graph looks weird. Sorry about that, it's fuzzy. So based on other studies, the pH content that we found in these hatch eggs are above levels we know to have sublytic effects in embryos, which means potentially affecting future GA classes. And I got extra funds to keep going these analysis, and now we're constructing the time series for the unhatched eggs, and also improving these pre-spill value. And this is very important because now that we're putting together data with the other collaborators in the team, the models have shown that there is a dramatic reduction in the abundance of biomass, the abundance and the biomass of deep pelagic organisms, about 50 to 80% since the spill, or since 2011. So here I'm showing this graph. This is the relative abundance, and on the bottom is showing the depths, 5 different depths in the water column. And here is their abundance in red in 2011, and in blue is 2015 and 16. So there's these very dramatic reductions that unfortunately we don't have pre-spill values, so we cannot specifically say is the water horizon. The chemistry kind of explain as a possibility of what is happening with the future generation of species, but also because we don't have any other data, this could be just 2011 could be just a very good year. And then we don't know the cycles at depth, so probably then 2050 is bad, and probably in 2 years it's going to be good or back or even better. So basically we need a future assessment in order to keep doing this work and to understand if there are some cycles or actually this is something that happened after the deep water horizon spill. So this is just to summarize our accomplishments in the different projects. And if you kind of notice from the first study to the end, it kind of like fits a little piece of the puzzle to understand what is going on at depth. And we still have a lot of questions, but so far we have been able to characterize chemically under different time and spatial scales what happens at depth. And we have followed this in time series for animals, identification not only organic molecules coming from oil spills, but as well from natural sources, in order to create geochemical indicators of ecosystem change, but as well sources, natural sources in the environment. And with that, as well, to increase our understanding of the fate of organic compounds in the marine system in order to make better assessment in the future. So with that, this has been my work, some of my work that I have been doing here at USF. And now I want to talk about a little bit about the future opportunities here. And so recently, as Amelia mentioned, I got a new opportunity to continue these studies on the deep pelagic ecosystem. And we're very excited because, as I mentioned, these trends we don't understand very well if it's actually the water horizon, or we're looking at some cycles that we don't understand in this deep pelagic ecosystem. And we're very excited because the program will enable us to keep doing our work for 5 up to 10 years. And so my role in this project will be to continue doing contaminants, but now we're not going to look only for specifically the oil spill, we're going to look for other contaminants, because there's a possibility the other stuff that have been accumulated at depth for many, many years probably could be the drivers for the changes that we have been observed. But also, this time I'm going to have the possibility to look at the water column, which I think we haven't paid that much attention. We haven't, because after, like in 2011, I showed the profiles, people says everything's gone, the water column is clean again. But to keep seeing the signature of the spill in the animals after 6 years of the spill, it means it's still there. And we're not the only group saying it's still out there. Brad Rossman as well have a year ago a paper that they found looking at C14 actually in the dissolved organic carbon pool at depth in the northern Gulf of Mexico. They found signature of the oil still there. So there's a lot of going on in the water column that we don't understand. So I'm hopefully going to be able to collect a lot of samples to look at POC or DOC. So there's a lot of opportunity for collaborations. And the rain comes. A lot of possibilities for collaborations here to look at if microbial communities actually can affect these pools, other contaminants, or other measurements that if people want to look into it, we're going to be able to collect a lot of samples over this time. So there's a lot of opportunity here to work. Also, a second opportunity that I recently got is to look at biosignatures and environmental conditions. And this is not only oil spill, this is anything that you want, and this diagram is just representing that. And you can look at counts, levels, concentrations, temporal spatial gradient, but you can choose different species or different contaminants, and can you use the same exercise to understand, and you can use signatures of processes or signatures of contamination events in the environment. So what we're doing now, we're testing, we're trying to apply molecules that haven't applied before to look at specific contaminations events, and we have some preliminary results that indicate that we're kind of good track for this. But this is as well open for anyone in the college if they want or are interested in a specific molecule that they would like to test it. We can do it, and we can develop the method. Also, we're testing at very different fauna, from shallow to deep systems, and this is very exciting because we're trying a lot of different things. And again, there's a space for collaborations. If someone is studying a specific group and they would like to be tested, we can do that in the lab. So with this, I just want to acknowledge that research like this cannot be done by one group. There's a lot of collaborations, but for sure I know I have missed an institution there. But most important part of doing research is the human part. And I've been super lucky to be part of very amazing groups. And there's a special thank I could stay the whole night here telling everyone how grateful I am for this experience, and especially Steve, which is director of CMS, Dave Hollander, Cheryl Gilbert, but also the technicians in the lab. So you probably don't know, but some of the students that are now students right now, and Bridget and Thea, they were before my technicians. Bridget were half-time, but I keep saying she was my technician. And they came after graduating from Eckerd College, and we trained them, and we gave them an opportunity to learn chemistry, but also to be part of a team and learn what is to be a scientist. And now they're doing their master, so I'm very excited about that. And also now we have a new text, which is Hannah and Olivia. So if you see them in the corridor, please say hello to her, to them. And thank you very much for being here on the rainy Friday before Halloween. And I'm open for questions.